So welcome back to Astrobiology Journal Club for the second installment of this term. Today we have Adrian Forsythe giving a talk on the origins of oxygenic photosynthesis and aerobic respiration. So welcome everyone back live attending on SaganNet, and take a look.
Oh, thanks, man. Okay, thanks for the introduction. I'm going to do my best to summarize this paper and not delve too much into the jargon because it's often really confusing, explaining these different kind of complexes and systems that a lot of cyanobacteria and photosynthesizing plants even use. And it's kind of a part of a larger body of literature and a larger, much deeper, interesting question that I think would be pretty great for us to discuss afterwards. Okay, so this is a paper from last year by Su et al., published in March 2017. And so we know that there's this great oxidation event that occurred, sometime between 2.4 to 2.1 billion years ago. We have evidence of this in the geological record, and there's about two competing models as to how this oxidation event took place. I'm going to quote from literature here because they do a much better job at summarizing them than I do. So they have the kind of initial, earlier two-step model in which the implication is that oxygen in the atmosphere remained well below modern levels until it rose to something more like modern values around 600 million years ago. And then in this view, we have kind of like a second O2 influx, which oxygenated much of the deep ocean, while also enriching the surface waters, and then thus kind of welcoming like other taxa to then populate land masses. Okay, and then competing with this is the more kind of contemporary, more integrative emergent model, in which after the great oxidation event, we really are asserting that oxygen rose again and then fell in the atmosphere and remained relatively, with relatively minor exceptions, pretty extremely low for more than a billion years or so. And then there's kind of this prolonged stasis, which is probably due to a combination of these biochemical kind of feedback loops. It gets all very complicated. But anyway, this ends with saying that pretty much the concentrations of bioessential elements are really sufficiently large in the Antarctic Ocean. And then with low oxygen availability, really the challenge of the emergence and diversification of other forms is really, anyway, that's the emergent model, is that we've got kind of this large point in time in which we have diversification happening after the great oxidation event as well. So this is more of the contemporary model in which we have this kind of really controversial question, which is when O2 producing photosynthesis really started occurring. So there's many people agree on when the great oxidation event occurred, but nobody really knows when O2 producing photosynthesis occurred. Really, this is the source of essentially all O2 in the atmosphere. We're going to talk about systems involving photosynthesis, electron transport chain, and then kind of this, which is kind of integrating these light-sensitive reactions to cellular respiration cycles. So there's evidence that points to various periods in history which can range of 1.5 million years. So it's all very contentious. There's sources of geological, geochemical, and paleontological evidence that kind of support different directions. And ultimately, this profoundly altered the course of evolution for life as we know it. This probably facilitated the development of really complex metabolisms and even probably complex multicellular life as well. So going from stuff that we kind of commonly associate with cyanobacteria to stuff that's more and more complex and organized. And so we often kind of associate the culprit of this production of oxygen in the atmosphere with what we know as cyanobacteria, right? This is commonly how we see them in these stromatolites. And really there's no other known processes that could have resulted in this large, massive increase in O2 levels. And really that this chlorophyll-based system originated within the bacterial domain. So we're talking about very early forms of life here, in which we have this introduction of this new kind of system involving photosynthesis and then spread across the domain, which is really common for bacteria to do using horizontal gene transfer. Yeah.
Adrian, what is it about aerobic respiration and oxygenic photosynthesis that can lead to more complex eukaryotic organisms? Could it not happen with anoxygenic photosynthesis or any other form of converting energy?
I think it's just a lot more efficient than other forms, based on what's readily available. Yeah, I think it's just it was the most efficient way for there to turn pretty much energy from sunlight to sugar and stuff. Although that probably could be contentious and is probably a point of discussion, but as I know of it, I don't see why not. Okay, cool. All right, moving on. Right, so we don't know exactly when photosynthesis was kind of this key point, or this place, when it first occurred in these cyanobacterial ancestors. Yes, so if this was true, if all cyanobacteria were photosynthesizing, we should see these genes for these specific complexes. I'm going to refer to complexes kind of 3 to 4, but I'm not going to go into great detail, mostly because I'm not super familiar with them. But pretty much there's different systems for kind of taking the ATP generated from photosynthesis, turning it into sugar in the cell. That's due to these specific complexes. So yes, if this is true, if all cyanobacteria as we know them should be photosynthesizing, we should see these genes responsible for these processes within, like across the taxon, because there's multiple phylums that make up cyanobacteria as a whole. Alternatively, if we consider that photosynthesis and aerobic respiration become evolved after this split, so they started just this new system after this diversification happened, we would kind of correlate this with the rise in oxygen levels. So it's kind of this correlative causative dichotomy that we're going to be talking about. Yeah, and then some other parts associated with this metabolism was kind of independently evolved, and we should see a lot of diversity between these complexes. Okay, so we're kind of they set this up as kind of testing either of these hypotheses. And so the way that they did that was through, I'm just kind of calling this exploratory genomic analysis, but pretty much what they were doing is they're mining publicly available data sets, which people have already done the sequencing for them, and they were looking for representatives of Cyanobacterium. So within Cyanobacterium, there's 3 lineages. One is the Oxyphotobacteria, which are kind of the main photosynthetic component of that group. And then there's 2 other ones, lessly kind of that are less represented and less well known. So really, they were trying to determine what really the components of the metabolism of these groups are, because they actually can't really be cultured and studied in lab. And so that's kind of like this main problem with a lot of genome sequencing and a lot of microbiology in lab, is that there's a vast diversity of life that we can't actually culture and can't actually study. And it's known as this kind of like a great plate count anomaly. So if you have kind of this sample from the environment, looking at the microbes, the diversity in particular, really only get 1% of those that can actually be cultured in lab, and sometimes it's very hard to determine what these components are in order for us to study them closer. So this kind of skips that problem, because we can isolate environmental DNA, we can sequence that environmental DNA, we can compare it. And that's pretty much what the workflow of metagenomic stuff is. So going from an environmental sample, they didn't sample viruses, but this is just the best graphic of metagenomic sequencing that I can find. Isolating and pretty much sequencing these DNA fragments in high contiguity. And there's a lot of replicates of these, right? So it's kind of binning them and pairing them and comparing them based on the specific sequences that we're interested in. And what's really interesting is that these are previously uncharacterized lineages. So we have no idea exactly what their metabolism consists of, is what I said before. But they knew they were out there based on some other environmental surveys. So they're kind of delving deeper into these unknown lineages of cyanobacteria. So the two that they specifically addressed in this paper are called Melainabacteria and Cytochromatia. So the first, Melainabacteria, there's not much I can say about it, but they found at least 28 complete genomes within this metagenomic public available data from various sources. So anywhere from the human gut all the way to the deep subsurface and aquatic environments as well, and wastewater. And here's a photo of what it looks like on the plate if you're interested. Not too interesting, but what's more interesting is this new phyla that they were able to really describe. Previous to this, it was only described as this nice jumble of letters and numbers, but they actually termed it in this paper from the Latin phrase late, right, just a Latin kind of amalgamation of words, late cytochrome cell color class. So it's really just this late arrival that they said to this class of Cyanobacterium involving this new cytochrome that they're going to talk about. Yeah, and they were able to actually assemble 3 draft genomes of this, which is really impressive for working with metagenomic sequencing. And what else? Yeah, they were recovered from these kind of photic and aphotic environments. So probably involving a lot of diversity in how they undergo photosynthesis that they do, or how they undergo respiration. Okay. This is a photo of Oxyphotobacteria, because there's no actual microscopy of members of this class yet, but although it probably would look something very similar to this. Okay, so based on this analysis, they really have determined that only members of Oxyphotobacteria are capable of oxygenic photosynthesis. So that's this file up here at the top. So the triangle here is showing like a lot of diversity within this class. Although other kind of phyla carry out simpler forms of photosynthesis, they just don't end up with, or they don't produce O2, so they're not characterized as oxygenic photosynthesis, which is what we're interested in here in this paper. And really that select members of all groups carry out some form of cellular respiration, although there's variations on these forms. And really that all cyanobacteria, so all 3 phyla in this group, have some shared characteristic of the electron transport chain and some variation of this motif. And of course, we get kind of these components lost, added over time. Right, so converse to what I was saying before, the series Cytochromatia and the Melainabacteria don't contain these genes for photosynthesis or carbon fixation. So that's lacking this green star and the green hexagon. Yeah, up here that's characteristic of the Oxyphotobacteria. This really suggests that the last common ancestors of Cyanobacteria were non-photosynthesizing in nature. And really that they gain this ability perhaps through horizontal gene transfer or some other kind of mechanism which isn't well described in this paper, but it's obviously a very contentious topic for future research. So this is really not consistent with the selective loss of all phyla having this ability to photosynthesize and then losing it. Or just, yeah, which indicates a cyanobacterial origins for phytotrophy, for oxygenic photosynthesis in general. And so this idea of horizontal gene transfer is pretty common, commonly talked about in most of the domains of life. It's very common, apparently, in bacteria. A common example is symbiotic theory, although it's not truly horizontal gene transfer, but that's kind of the basic idea. And so really there's this idea that both the photosynthetic ability and has horizontally transferred across taxa multiple times compared to vertical gene transfer, which is just kind of this descent through a phylogeny in time. So that it's occurred for photosynthesis in a lot of the Oxyphotobacteria, but also within the Melania and series Cytochromatia for aerobic respiration, in which they try and describe in this paper, although it gets very heavy on the jargon, which I will go through a bit of it now. Just the most interesting findings that they were talking about was really that there's this independent evolution of aerobic respiration, postulated to be caused through horizontal tree transfer. And so really within these two groups, in Melainabacteria and Cytochromatia, there's kind of these two evolutionarily unrelated complexes that are involved in aerobic respiration kind of cascade. And in some cases, they're really specific to the environments that these microbes are found in. So they evolve under high and low oxygen levels, and there's specific kind of components to those environments. And there's also, like I said, adaptation to in this complex form, in complex 3. Okay. Yes, so continuing kind of on this theme of this divergence and this independent evolution of aerobic respiration, there's also this very interesting, what they call a fusion of these complexes in some specific cases, although it's really not a fusion per se, because that would indicate there's some shared ancestry, although it's very independent. So there's this kind of like, this independent evolution of this kind of combined complexes in this case. So in this specific case in Melainabacteria, we've got C family oxygen reduxases and BC complexes kind of stuck together. So it's kind of doing the jobs of these two complexes as kind of one unit compared to these two different complexes, which usually work in tandem or work in conjunction with each other in the respiration cycle. And yeah, it's specific to this group. There's a lot more diversity than we get with the other phyla of cyanobacteria, which is also interesting. So I didn't want to delve too far into it, so I'm just going to I'm going to talk about some of the main conclusions from these results instead of going deeper into it. So really, it's that the genomic data here only supports an hypothesis in which this oxygenic photosynthesis occurred later, or arose later in bacterial evolution than what was previously thought. So we really thought that cyanobacteria were coming kind of out of the gate, photosynthesizing already, causing this great oxidation event. And really this is inconsistent with this idea that they were really kind of like the progenitors of this specific form of photosynthesis, and really that oxygenic photosynthesis is a derived feature of a specific lineage of cyanobacteria. So there was this specific kind of time in which cyanobacteria were not all photosynthesizing. And really the maximum age at which this oxygenic photosynthesis probably occurred was around the divergence of these two lineages that they were talking about in this paper, the Melania and the Oxyphotobacteria, actually around 2.5, 2.6 billion years ago. And so consistent with this time period, there's geochemical evidence that points to that before the rise of oxygen, there was manganese oxidizing phototrophy. And so this is really considered to be the direct precursor to oxygenic photosynthesis. And further, this suggests that this form of photosynthesis was really kind of coinciding with the rise of the great oxygen event, with the rise of atmospheric O2 around 2.3 billion years ago. So if the last common ancestor of cyanobacteria did not use oxygen, this really suggests that there was insufficient oxygen until the appearance produced of this atmospheric oxygen produced by photosynthesis. And so alternatively, it's kind of this part of this new model of how the great oxygen event occurred, that there was this manganese oxidizing kind of part of life, in which we've got, which again is consistent with the times of the divergence of these two phyla, and what else? There's a lot of, this is supported by geochemical evidence as well. And so if I can kind of draw a parallel with some astrobiological significance, obviously we know now that the early environment of Mars was much warmer, wetter, and probably more oxidizing than the current day, and obviously most likely oxygenated as well. And so we have these records of Martian strata, which has really recorded these biochemical flux, or shouldn't say biochemical, but these chemical fluxes of oxygen and other components that could be due to something like a photochemical process. This is similar, but not completely parallel with what happens on early Earth, in which we have these sedimentary rocks on Mars with manganese and oxychlorine, kind of rich components to them, which would be really important for an early oxygen cycle. Yeah, and so in comparison, really Earth did not develop these enrichments until after this great oxidation event occurred. And so a lot of this evidence is from the crater and mountain sharp, as shown here. I think that's actually it. I'm just going to wrap up there, and really be happy to take any